Alright, welcome to the Unit 5 tutorial. Unit 5 starts out with talking about some of the fibrous proteins, and then a big discussion about myoglobin and hemoglobin and how those proteins work to transport oxygen and CO2 in the blood. So there's a lot of material, 95 slides, so I'm going to move pretty quickly through these and just hit the main points. Some of the concepts with hemoglobin, especially in the binding, is complex. So Dr. Bracho, and you can learn it better in SI. So I'm just going to touch on what you should really know definitely for the tests. The second time you've seen this chart in a row, that means it's important. Make sure you know this well. This chart is going to be described kind of as we go through the three main fibrous. The first one is keratin. You should know where it is in the body, any amino acids associated with it, and then the general structure. So F-keratin is almost entirely alpha helical. So in the last one I said myoglobin is a big one, because myoglobin is like 90%, or sorry, not 90, about 78% alpha helixes, 0% beta sheets, which means about 22% turns and loops. So myoglobin is very high in alpha helixes, but this one is like 100% almost alpha helical. So keratin would win if it was asked which one has the highest proportion of alpha helix. I would go with keratin, and if keratin's not there, maybe myoglobin or collagen. We'll look at collagen in a minute. Here we go. So most abundant protein, that might come up. It's going to be the largest one we talk about. Extracellular matrix, skin and bones, provides strength, good things to know. And it's not a regular alpha helix, it's a like triple helical alpha helix. So knowing the details of it, so we're going to rank collagen as the heaviest one that we talk about in this unit. Lots of glycine and proline. It's also rich in hydroxylysine. Hydroxy, remember, is just an OH group. So OH, lyse, and hydroxyproline will be how it's written out. Make sure to know collagen has both. We're going to look at elastin. It's only going to have one of the two. Collagen is glycosylated, which means there's sugars attached to the proteins. Those are the sugars. It's the only one that has carbohydrates, sugars, any oses attached to it. So the structure, a little less important, but definitely know that there are cross-links happening, and the cross-links happening with lysine and hydroxylysine, and any version of them. Yeah, don't get caught up in trying to understand too much of those. This might be like a quiz level thing for the types of cartilages, collagens. I was looking at the word cartilage here. What I would, if I was learning this, I wouldn't worry about anything in these columns. I would know type 1, 2, and 3 and where they're found. And then I would associate 1 and 5 are basically the same, and 6 and 2 are very similar as well. So that was kind of the schematic of this graph I had in my head. Type 1 is your most abundant one. But they're all the alpha triple helical. They're all essentially the same properties, so don't let him confuse you by saying type 1, 2, or type 3 collagen. As far as size or anything like that goes, it's all essentially the same. It's just the distribution of where it is in the body that's different. So these enzymes, when you're hydroxylating collagen, which means adding these here, it's done by these enzymes. So these are globular enzymes, and they are, ascorbate is vitamin C, so they're vitamin C dependent. You don't really need to worry about the reactions. So he's going to label these. Make sure you can say which happens in cells versus out of cells. Which ones are enzyme catalyzed and spontaneous. Basically just know this whole chart really well. So these are the things that happen inside the cells. And then these are the things that happen outside the cells. So remember, cross-links done by lysyl oxidase versus, not to be confused with lysyl hydroxylase, which was this enzyme. Lysyl hydroxylase outside the cells for hydroxylation. Lysyl oxidase, cross-link formation outside the cells. This is how it's done. If you understand it, great. It's not going to be tested over, most likely. I've never seen a question over it. Just know that lysyl oxidase does the cross-links outside the cells. A lot of clinical stuff in this unit relative to the other units, so make sure you know type 1, 2, and 3 collagen diseases. Elastin, it tends to be the second biggest at 75,000. So no sugars attached. It has some hydroxyproline, but no hydroxylysine. So make sure you know that part. So it'll still have the enzyme prolyl hydroxylase, but it will not have any lysyl hydroxylase. None of that. It's found in tissues. Mostly the aorta is his favorite example. So while it doesn't have any hydroxylysine, it still is cross-linked by lysines. Desmosine is a word you might want to associate with elastin as well. So those are your big three fibrous proteins. Know them really well. Many questions will come up with them. So catalase, we're talking about some heme proteins now. 4 heme proteins we're going to discuss. The first 2, this is all we're going to see about them. Heme meaning they have an iron attached, which you might say a mineral in a test, or inorganic molecule. Any of those could be talking about iron. Catalase is the fastest, breaking down hydrogen peroxide. Cytochrome C is used for energy, and oxidative phosphorylation used to make O2. These are both redox enzymes. The other 2 heme proteins are myoglobin and hemoglobin. So myoglobin muscles, you store it there. Hemoglobin is obviously your red blood cell oxygen transport. So, myoglobin is muscles. Also, you store heart and liver, the other very important places in your body. And they both bind oxygen reversibly. There's going to be a few major differences that we're going to talk about later in the unit. Hemoglobin, red blood cells only. So this is the structure of a heme. It's an iron attached to all these nitrogens and then all these other rings. And you have a histidine attached to the iron, which is represented here, and then that's where you carry the oxygen, O2. Don't worry too much about the structure, but you should know this guy. Be able to write this out in order. There are some tests where he'll just it'll all look like this, and he'll just mix up a few things, mix up the order. So the big takeaways for a quiz, definitely, I could see him asking something like that. For a test, it's usually knowing the oxygen binds between helix F and helix E. Myoglobin is relatively smaller, only about 17,000. contains one heme group, so make sure you can do a good compare and contrast myoglobin versus hemoglobin. I already talked about this point here. 78% alpha helical. Big takeaway on this one is what's in red. The binding of oxygen is simple. So simple meaning it's only dependent on the concentration of myoglobin, which is abbreviated MB. Oops, there it is. And the pressure of oxygen, so the amount of oxygen available. This never comes up. It's a little hard to explain, so it'll make a little more sense in biochem too. So don't worry too much about that now. Same thing with this. We'll talk about this in the next unit. If you understand it, great. It's just a little complex, and I'm not going to go into it in the video, and it's not going to be tested over that much. I'll show you what's tested over. It's mainly with hemoglobin. So this is just showing that binding happens very quickly with myoglobin. Don't let this stuff freak you out. So carbon monoxide, we're all familiar with carbon monoxide poisoning. It binds much stronger than oxygen does. So a little bit of carbon monoxide can cause a big problem with people. That's the greater affinity, so the carbon monoxide, the CO, is going to get in there and bind much tighter than the oxygen does, and it's not going to let go. So hemoglobin kind of looks like four myoglobins stuck together. Two that are very similar and two that are pretty similar. It's two alpha globins and two beta globins are all what makes up the myoglobin unit. So it's about 4 times as big, approximately. If we're ranking things so far, we know we got collagen at 100,000. We know we have elastin at 75,000 and hemoglobin at 64,500. Then there's a huge gap, insulin, myoglobin, they're all super small. Keratin is so inconsistent, so it's not generally used. So I would know these as your big three as far as size goes. Back to hemoglobin, it has 4 heme groups, so 4 iron. This will be a common abbreviation, or just HB, for hemoglobin. So we've already said it's a transporter, it's red blood cells only, it's complex, it's dependent on many things. We're going to look at that in a much later slide called the Bohr effect. And there are different types of it, so there's a fetal version of it that behaves differently. We talk about the adult hemoglobin, HbA, which is most of the hemoglobin in the blood of the adults. Big point here, we'll see on a graph, fetal hemoglobin binds O2 greater than adult hemoglobin, which just means it can stick, it can grab the oxygen faster, and it's going to let it go at a lower rate. For adults, it won't stick as well, but you can get rid of it and transport it. You can release it much better. So cooperatively, it's going to talk about the domino effect. Meaning you have 4 positions here. Once you get one O2, it makes it easier for the next one. Once you get these 2, it's even easier for the third one. And then by that time, it's super easy to grab the 4th. O2 only has, myoglobin only has 1, so it has a greater affinity, binding it too tightly, so it's only good for storage. And then it works backwards as well. That's what this statement's saying. So once I get rid of one, then the others just fall down like dominoes. Nice and easy. There's not much questions over this stuff. all the questions generally are going to be about the R and T states. No about 2,3-BPG. I won't go through it, but there might be some questions in there. But what I'm looking for, here's more 2,3-BPG for you to review. Just good to know these slides. It's a little too much to go into in this short video here. I'm going to come back to fetal hemoglobin. What I'm looking for is the Bohr effect, which is where most of these questions come from. So on these two slides, know that R form means oxygen, and these are the 5 things to know about it. If you have more oxygen here, you have less carbon dioxide. If you have less protons, you go more basic, your pH goes up. In both situations, 2,3-BPG concentration does not affect it. So if you know one, then you automatically know the other, because it's the same exact thing, except the first 4 arrows reversed. So that's going to be most of the questions on how we'll ask it for this binding. So hemoglobin is very similar. We just said it binds a little bit tighter because it has less basic amino acids, less positive charge, so less 2,3-BPG. 2,3-BPG, you'll learn, facilitates the loading and unloading process. This is good to know about, especially the top 2. So the adult hemoglobin is most of it. You have a trace amount of fetal hemoglobin, and you have a couple other specialty type hemoglobins as well. I would be able to draw these out because he asks a lot of things. So we have conception right here, time of birth right here. So you have alpha hemoglobin is turned on at the time of birth and takes off. You have this guy, which I don't know the letter of it. It starts out, that's the only hemoglobin you have, but it goes away pretty early on. It goes away in the first trimester. A popular one is beta hemoglobin, beta here is turned on at birth, but it doesn't increase to significant amounts until after birth. Turned on at conception, significant amounts after birth. This guy here is turned on right before conception. And then the fetal one, turned on at birth and then significantly decreases after conception. So be able to tell the stories of especially alpha, beta, but I definitely recommend these other 3 too. If you've memorized all that, you might as well memorize the last one as well. So he does ask questions off of that graph. This is kind of extra information, I think. I haven't seen questions on it. So more about carbon monoxide. Stabilizes the R form, so you can't get rid of the oxygen you do have. So it hits you on a couple levels. So this is explaining the Bohr effect that we already mentioned here. Just know the direction of the arrows. If you know all of them, even without knowing the question, you're going to have all of them look exactly the same like this, except you might have something like this. And it's kind of like playing odd man out at that point. I mean, you should definitely understand the concept, but make sure you have your Bohr effect arrows memorized for R and T states. We'll talk about sickle cell, another clinical thing. He often calls it a single amino acid substitution because you're supposed to have glutamate, but instead you have valine. So you go from a acidic charged amino acid to a nonpolar. Big difference. If you just have genetically one of them, it's not that bad. Not significantly affected, resistant to malaria. If you have both of them, then it's very dangerous. but single amino acid substitution or something like that is usually how he phrases this one, or disease of hemoglobin versus all the collagen diseases. Let's see, there's a few more clinical things. Be familiar with these. These are also globin diseases as opposed to the collagen diseases. And that's it for Unit 5.